bang dot. Yeah, it's from the back, so you go. In research, there's no path which somebody tells you to go, especially in those parts of research which are unknown, and that's often the best. And so there you have to pick the right direction. You sure of this?
As I understand from Stefan, yeah.
Reinhard really has an amazing vision for where to go and what to do.
This is Reinhard Genzel and his team of astronomers from the Max Planck Institute for Extraterrestrial Physics near Munich. Today they're at leisure, hiking in southern Bavaria.
I make jokes that our PhD students ought to have been on a mountain before they finish, but that's certainly not required. Having a team of good people is absolutely essential.
Genzel and his team are credited with demonstrating that at the middle of the Milky Way, the galactic center, there is a supermassive black hole.
Our measurements in the end now are so close in that we can exclude any other configuration but a black hole. My interest in the galactic center started already about 28 years ago.
He was at the University of California at Berkeley then. Led by his mentor Charles Townes, the team there had already theorized there was something massive at the center of the Milky Way, maybe a black hole.
So the galactic center is somewhere in there, but unfortunately, between us and the Milky Way center, and that's 25,000 light years, is a lot of interstellar material. And you see this on this picture as these dark splotches. That's dust.
Astronomers have to overcome that dust, as well as the Earth's atmosphere, which blurs images. Genzel's team went to high-altitude conditions in the Atacama Desert in northern Chile. This is where the European Southern Observatory operates the so-called Very Large Telescope, or VLT. Actually, it's 4 telescopes.
There we have the best conditions to do our research with very big telescopes, 8 meters in diameter.
The telescopes can be set to see light at infrared wavelengths. Most light in space cannot be seen by the naked eye. Here's an example. This is Orion's belt seen in visible light. Then the same picture again, and gases not visible before show up in the infrared. At the center of the galaxy, the infrared neutralizes interstellar dust.
With the infrared, we can peer through this dust and make our measurements. Also in the infrared, we can use then the techniques of adaptive optics.
Adaptive optics was developed at the end of the Cold War by the U.S. military so that it could see more clearly in space.
If the Earth's atmosphere acts on the incoming waves of light coming into they bend it. So anything which was straight all of a sudden is wiggly. And the technique is to measure that wiggliness and then undo it through a so-called deformable mirror.
Here's the effect. This is the galactic center without adaptive optics.
Now switch on the adaptive optics, and all of a sudden your image sharpness is better by a factor of 20 or so. The astronomy has here, as it has before, benefited from applications in the military sector, which we now can use for these pure basic research efforts.
Genzel's team built a device for the Very Large Telescope in Chile that has adaptive optics. It gave them clearer pictures of the galactic center.
Yes, and that was really the key of the success. Make sharp enough images, beat the Earth atmosphere, which distorts the images, etc.
And there was some luck. Seeing more clearly meant finding a star not visible before. It showed the Genzel team there really was a black hole.
Star S2, we were extremely lucky, and nature gave to us, so to speak, a gift.
For 10 years, from 1992, they tracked S2, starting here at the top, when it was moving slowly, then all the way down. When it got to the bottom, it picked up speed as it went around the Sagittarius A star, identified as the best candidate for the black hole. The increased speed of S2 at that point confirmed that it was in a classic orbit of something massive.
The planets which surround the Sun, when they're very far away, go very slow. A planet which is closer by has to go more quickly. That's a basic result of gravity theory of Newton and also Einstein, of course.
The orbits of around 30 stars have now been traced around the black hole.
Nobody would have expected in the galactic center that there are stars moving around a black hole on time scale which you and I would call normal human lifetime scales. And so we surely couldn't hope that, but it was the case.
Genzel's team has been busy with more instruments. Sometime next year, the Infrared Space Observatory Herschel is due to be launched. Max Planck has built a spectrometer, which will collect the light that comes into the telescope on Herschel. It's also built a photodetector array, which will analyze the properties of the light.
This took us 10 years because it has never been built before. The whole instrument, starting from the detectors, the whole design, the material, etc., was a completely new development.
Every member of Reinhard Genzel's family has taken up science. His wife is Orsolia Genzel-Boroviceni, a pediatrician specializing in neonatology at a women's hospital in Munich.
I particularly take care of very small premature babies or babies who have trouble around birth and need to go on our intensive care unit.
When we visited, one of the Genzel's two daughters, Lisa, was writing exams on her way to becoming a medical doctor. The other daughter, Daria, stopped by to show her mother a new dog.
I studied biology and did my PhD in the behavior and training of bats. Now I'm working as a dog trainer, mainly in the animal pound. And my main work there is to train dogs which have been very aggressive towards humans or towards other animals or towards dogs.
Here you can pick what you want to eat. I'll take a soup. Cheers.
Reinhard and Orsolia Genzel-Boroviceni put in long hours doing jobs they're passionate about.
It's cold, yeah. I think the kids are like that too because that's what they see around them. You know, they've always seen us working, and even when they were little, my husband always took the work with him in vacation.
The Shaw Prize is not Reinhard Genzel's first award. In the past, though, he was slow to tell his family.
My family upbringing was do not brag. But I got into trouble the last time, so I guess what you heard was that I'm behaving a little better.
He's learned now that he has to tell me first because he once won a prize and I found out by a crazy person calling me at night. Our phone number is not listed anymore because people call and say they have been abducted by extraterrestrials and have seen the black hole, or, as I said, claim that had stolen my husband has stolen the black hole, like literally taken it and stolen it.
Yeah, that happened once before. Yes, black holes, apparently create quite a variety of responses.
Reinhard Genzel still spends about a quarter of his time working as a professor at Berkeley in California. Mainly, he's scientific director at Max Planck, which has some 80 institutes in Germany, including 3 near this one in Garching. Genzel likes the long-term funding here that's made special work possible.
Identify the key leaders who are likely going to bring that field, especially in innovative new fields, forward. And then let them research, no strings attached almost. It's a paradise.
The work Wilmot is doing now is based on a breakthrough by Japanese researcher Shinya Yamanaka. Yamanaka had seen the potential of using cloning to produce stem cells for research and human therapies.
Nuclear transfer is a very good way to make stem cells from patients' own cells. But technically, it's very challenging. It's very difficult.
And it still hasn't actually been done. South Korean researcher Hwang Woo-suk was outed as a fraud 2 years ago for saying he had created human embryo stem cells by cloning when he had not. And Yamanaka says he has qualms about taking embryo stem cells, sometimes known as ES cells, because the embryo is destroyed in the process. He'd seen one through a microscope at a fertility clinic.
I did feel that it is a very special cell. It's not just regular cell. You know, I have 2 daughters, and the differences between my daughters and those human embryos are very small, actually. If human ES cells or human embryo, using human embryos is the only way to save the patients, I would be happy to do that. At the same time, we have to do our best to avoid such a usage of human embryos.
In his work at the University of California in San Francisco and at Kyoto University in Japan, Yamanaka searched for another way to get stem cells. His first breakthrough came 2 years ago. He found 4 genes that, when added to the skin cells of adult mice, caused the cells to reverse and become like stem cells again. Genes give instructions to cells so they can produce proteins. Yamanaka called these new cells induced pluripotent cells, or iPS cells.
It is a breakthrough in that we only need 3 or 4 factors to induce reprogramming. So it's very easy.
Every cell in a person's body has DNA unique to that person. Skin cells, blood cells, liver cells, all share the same 20,000 or so genes. During human development in the embryo stage, different genes in different cells get switched on and off in different ways. At that time, the different cell types are created in the body. It's called cell programming. Yamanaka thought that if he could find the gene switches responsible for programming stem cells, he could flip those same switches in individual adult cells, like skin cells, and reprogram them so they return to their original stem cell-like stage. But how could he find the right genes out of some 20,000 possibilities? He used mice for his research and made a bold hypothesis.
We hypothesized that factors that induce pluripotencies are largely overlapped with factors that maintain pluripotency in mouse ear cells. So based on that hypothesis, we decided to start a project in which we try to identify factors that maintain pluripotency in mouse ear cells.
Yamanaka put his team to work. Based on his hypothesis and through computer analysis, they found 100 most likely candidates from the pool of some 20,000 genes. Senior team member Kazutoshi Takahashi credits Yamanaka with delegating responsibility to his team so each member can explore his potential.
In many other labs, there is one boss and several sub-leaders reporting to the boss. However, such a vertical structure doesn't exist in our lab. Everyone has active interaction directly with Dr. Yamanaka.
After more than 3 years analyzing hundreds of thousands of cells, Yamanaka and his team narrowed the gene pool down to 24 and finally 4. He then got these 4 selected genes to make iPS cells. He took skin cells from an adult mouse, then used a virus as a means to insert the 4 genes inside the cells. 2 weeks later, the skin cells had completely transformed.
Probably the most success in my 20 years as a scientist.
Less than a year later, Yamanaka and U.S. researcher James Thompson, working independently, made headlines by publishing papers that said 4 different genes could be used to make human skin cells pluripotent.
Now we can generate stem cells directly from patients. And from those iPS cells, we can generate like cardiac cells or neural cells which have the same set of genes with patients. So those cells should be very, very useful in understanding why and how those patients became sick.
Experiments, though, with mice cloned from the iPS cells shortly after the breakthrough in 2006 showed one of the genes, c-Myc, was an oncogene. It caused cancer.
When we made new mice from those new stem cells, many of those mice developed tumors in their life.
Later, Yamanaka found c-Myc could be removed and only 3 genes were needed to reprogram skin cells to become stem cells.
So we don't have to use such a dangerous gene or a dangerous protein.
There are further challenges. The virus used to shuttle the specified 4 genes into skin cells, called a retrovirus, can mutate a patient's DNA and cause cancer.
They may be very useful for research, but for therapy, they've probably got big problems. So we've got to find a way of actually doing it without the viruses, probably.
I think it's just a technical issue. So in other words, it's just a matter of time. So in the near future, I think we or somebody else would identify ways to induce the same stem cells without the usage of retroviruses.
Before you could think of putting cells like this into people, the procedure needs to be modified so that there is no risk of it. And it may be that the kind of basic research we're carrying out can contribute to that.
This is Sir Ian Wilmot's lab at the University of Edinburgh's Centre for Regenerative Medicine. He'll use iPS cells in his new research.
The ability that Shinya Yamanaka established of being able to treat cells taken from an adult so that they become equivalent to embryo stem cells has completely revolutionized things generally, including for us.
Wilmot has started work on therapies for ALS, also known as motor neuron disease. It is invariably fatal.
It's a very unpleasant disease. Once a person has the symptoms, they'll slowly lose control of their muscles.
It has been many decades since I have been out of my wheelchair.
Scientist Stephen Hawking has had ALS for 45 years. He's among an estimated 10% of sufferers who survive more than 10 years. Wilmot will turn iPS cells into patients' nerve cells so he can see why they don't work properly.
ALS symptoms arise because of degeneration in the nerves that run down the spine. And so you can't directly study those and you can't take them from a patient. It's just not possible. So what research workers really want is to have cells which are equivalent to those in the patient.
This example in animal research shows how a lab rat can move again freely after human stem cells were used to repair a damaged spinal cord. But the world awaits the first human therapeutic use of stem cells, or iPS cells.
What the first therapy will be, I wouldn't like to say, and I wouldn't like to put a timeline on that, although obviously the pressure is on to get this as quickly as possible.
Ian Wilmot and Keith Campbell had shown they could reverse the differentiated cell, then reprogram it.
It proved that cells were not fixed in a particular specialised type. So your skin cell could somehow, or the genetic material in that skin cell, could be made to become a liver cell or a hair cell or whatever. Previously, it had been suggested and written by scientists that this was an impossible task. I don't believe anything's impossible. I think some things are improbable.
And cloning, in theory, could also produce human embryo stem cells, the sort that can become any cell type in the body. When the cloning breakthroughs were made, Keith Campbell was working at Roslin as a postdoctoral student. Keith now has his own lab at the University of Nottingham, well away from the main campus.
In relative countryside, and I think it's a great aid to thinking, clearing the head.
His team still uses nuclear transfer, the only technique to date that's been used for cloning mammals.
We're able to take one nucleus from an adult animal and place that into an egg. So we bypass, there's no fertilization.
First, a laser cuts through the zona pellucida, the outer shell of an unfertilized egg, or oocyte. Now they can go after the nucleus, the mother's DNA.
He then inserts the needle and is able to actually extract the maternal DNA.
The oocyte is then injected with the nucleus, the DNA from an adult cell of another animal.
A skin cell or any other cell in the body, which we know contains intact sequences of both the original paternal and maternal DNA. We pick it up and we go back through the hole we've made in the zona pellucida.
And the nucleus from the adult cell is put in contact with the oocyte. Development will only start after one last bit of intervention. The oocyte and the new nucleus sitting there on top, they're now called a couplet, are jolted to life.
A DC electric current will cause partial breakdown of the membrane.
And the oocyte and new nucleus will fuse together. The resulting embryo is grown for about 7 days in a dish, then put into a surrogate mother. Many early embryos weren't viable, though, because the oocyte and the new nucleus, both often called cells, were at different stages of their cell cycle.
The understanding that Keith brought to the project was about the mechanisms which control the way in which cells grow and divide, which are very complicated, and it's critical to control them.
Keith found a way to stop the growth of the cell being transferred to the oocyte. Before the DNA was copied or synthesized, the cell was brought to rest at a place called G0, within gap stage 1, where there is less development.
You can induce cells to enter this quiescent or G0 state simply by taking away some essential nutrients.
In 1997, 1 year after Dolly, the Roslin team cloned Polly. She was a transgenic sheep. She'd been given a human gene to show how animals can make what some people lack. Polly produced a blood clotting protein in her milk.
Once we've made the founder animals, we just breed them, and it's just a matter of extracting the protein. Oh, yeah, okay. Oh, do you want some of this? When I come down here, I'm looking at the quality of the animals. I mean, I'm interested, obviously, in all aspects of the process, from when we make them in the lab, through the pregnancy, to the birth, and then through the growth of the animals. We have 6 mothers and 7 lambs. One of the mothers had twins. Most of these animals had 2 or 3 embryos put into them.
Since the days at Roslin, Keith has been working on making cloning more efficient.
We've increased the frequency of embryos which will develop to live, viable, healthy offspring. We're now at about 20%. When we did Dolly, it was about 3.5%. It was 1 out of 29. We have some sheep made from adult cell lines, and then we have sheep made from 3 fetal cell lines.
Mice have long been used as models to treat human disease. Sheep, though, are more like humans. At Nottingham University, the sheep cloning program is tied to medical research.
An animal such as a sheep, there are some similarities in a number of areas, and that is why we felt that cloning in sheep was, certainly from animal models situation, could be even perceived as a better model than a mouse, for example.
Like Ian Wilmot, Keith Campbell traveled a lot because of the attention generated by Dolly. Some of the travel was for an American biomedical company that he later worked for.
The company had a base in the States, and I was asked to give talks at local schools, the local colleges, even the local churches on the implications of cloning.
Keith draws a line at cloning people.
I'm personally opposed to reproductive cloning because I think there are thousands of children out there at the moment that need adopting anyway.
Keith still travels to speak, but he tries to do less of it now.
I don't get paid just for going out and giving lectures. I get paid for doing research, teaching students.
Four of his PhD students, including Brian Woodward, graduated this year.
No matter what problems you've got, he's very approachable, laid-back guy, very supportive, very helpful, knows what he's talking about, is willing to share ideas, help you push your ideas.
Traveling and lecturing caused Keith to meet his wife, Kathy.
We met at a conference where Keith was a speaker.
Kathy was there as a representative for a U.S. company that makes lab equipment. They were married 4 years ago, and this year, she moved to England.
It started to become impossible to conduct our relationship from 3,000 miles apart and conduct two independent professional lives.
When there's time, Keith sneaks in some work on a car he's building in his garage. As a younger man, he had to know how to fix cars just to keep them running.
It became quite therapeutic. It takes my mind away from science, lets me do something with my hands.
The word mathematics means exact knowledge.
Vladimir Arnold majored in mechanics and mathematics at Moscow State University in the 1950s. In 1954, his mentor, Andrei Kolmogorov, outlined the KAM theorem, subsequently proved by Arnold and Jürgen Moser. It involved the stability of dynamical systems, such as two planets orbiting the Sun. They would interact and perturb the orbits. Theorems have been produced on this three-body problem.
The fact that forever the planets would not fall with each other, collide, and so on. But it's very difficult to prove. KAM theory is the first attack to this problem for 3 centuries, continued by thousands of different people in different domains, in mathematics, space research, celestial mechanics, astronomy, physics, thermonuclear reaction, magnetic field theory.
One example are the reflections from charged particles trapped in the strong polar magnetic fields, known as aurora borealis and aurora australis.
The question is, how long would it remain there? It's very similar to the thermonuclear problem. The KAM theory is providing also the solution of this. But I have done independent progress in an independent problem.
Take this huge TV antenna in Moscow.
You have a net of metallic rods. You start from a point and you go upstairs along one of those system of line. But then you descend the point A to the point B. If there is a resonance, then there might be a perturbation along this line. The measurements show that when there is a resonance, it is unstable. And the same technology used to study the three-body problem can be also used to study the stability of those things.
Arnold remembers mentor Kolmogorov for more than his maths. Students in Soviet times had their stipends rescinded for offending the Communist Party.
Kolmogorov told to this student, don't be afraid. I have a lot of money. I have paid you for my pocket 2 times more. And he has done it. His principle, the talent should be pardoned. I was pardoned every year, all the time. It was wonderful.
Arnold got his PhD and taught at Moscow State. At 18, still an undergrad, Arnold wrote a letter to the Soviet physicist and Nobel laureate Lev Landau.
I have read 10 volumes of your theoretical physics. I have discovered 199 mistakes, and he agreed with all of them.
The mistakes Arnold found were eventually corrected in the American edition of Landau's volumes.
I am maybe the last universal mathematician who is working in hundreds of different domains.
One celebrated Arnold conjecture is based on the work of French mathematician Henri Poincaré.
Consider a circular ring. Every point of A is sent to a new point. The conjecture of Poincaré. F preserves areas. Second condition. F rotates the boundary, which has 2 parts, oppositely. Poincaré conjecture. F has at least 2 fixed points.
Under these 2 conditions,
the science is called symplectic topology today. What I have done, I have extended it. For instance, I have considered the case of a torus surface. there is no boundary. However, I have invented the condition representing it. It's called exact mapping. They are not conjecture. It's not for the two-dimensional torus. It's for any manifold, any surface of any dimension.
Since 1986, Arnold has been with the Steklov Institute of Mathematics. His current work involves the mathematical theory of randomness.
The geometrical progressions are hundreds of times more random than the arithmetical progressions. So this problem, being random, not have random origin, neither the first nor the second, somewhere this problem of randomness is always presented.
Arnold lives in an apartment a few minutes' walk from the Maths Institute.
Here we are.
Do you play the piano?
No, I'm not playing, but my grandchildren are playing. I'm very old. I have grandchildren. Like that already.
Arnold is 71.
That's my father. He was a mathematician. This is my first mathematical work. I was 8 years old when I made this.
Several generations of Arnold's family have been scientists.
In the family, they told that my first mathematical discovery was made when I was 4 years old.
It was during the war. The family had a flat in Moscow in which the floor was very uneven. His father tried to place a stool, but it wouldn't stay.
And then I observed him, and I commented that you have moved the chair, turning it by a right angle already. And it never stayed on 4. This proves that the chair is wrong too.
Father then sawed the stool legs evenly.
My common life is like that.
Spent in the wild, swimming across rivers, skiing in the Alps.
For some 50 miles, bicycling, collecting mushrooms.
That's when mathematics came to him.
In places like that, sitting or even lying in the grass, I'm writing about 50 pages a day of mathematical calculations. It's a lot. My wife is displeased by that because she has not enough money to buy the paper. But that's her, my wife.
Eleonora is a medical doctor.
How did you two meet?
That's a result of a bicycling accident in the forest near the Moscow River. We married several years later.
You understand Vladimir's work, the maths?
No. She's honest. She would not pretend to understand it.
Is there something you want to do before you stop working?
I never stop. I have no plans to stop. No.
St. Petersburg, Russia. Native son, Professor Ludwig Fadeev, attended and taught at Leningrad, now St. Petersburg, State University.
All my family were at university.
Notably, his father, Dmitry Edvich Fadeev, was professor of mathematics.
I think that among young people there is feeling of independence, and so I really wanted to do what I can do myself.
So Ludwig chose physics instead.
This small red building says birthplace of theoretical physics in St. Petersburg, in Leningrad. 50 years ago, it was almost unknown.
Ludwig himself blazed trails in the early 1960s with his doctoral thesis in physical and mathematical sciences. It was related to the scattering theory of particles.
In the case of 3 particles, they could come on each other, scatter, collide, go back to infinity. So information about all possible processes in this system are included in what's called three-body problem.
It was a problem which stay in front of theoretical physicists, say, 30, 40 years.
Peter Kulish is a protege of Fadeev.
It was quite tricky to prove that now these equations are ready to be used.
In Faddeev's equations, the T matrix for the scattering, K and P for the momentum of three particles.
My contribution was in understanding that this particular operator must be taken as a sum of three different ones, which corresponds to three possible channels. At that time, nuclear physics was very much on the top. And so my paper was immediately used for practical calculations for nuclear reactors. I have about 200 published papers in journals and books.
It's said all computer calculations needed for application today use Faddeev's equations.
My doctor thesis gave me really international acknowledgement, but young Mills gave me more fame, so to say.
In the mid-50s, Yang Qianming and Robert Mills proposed a quantum field theory to resolve some confusion in particle physics.
Young Mills field itself was vector field, but also a matrix which acts on this. So it's many-dimensional object. But what are the particles, what are excitations which correspond to this field? I decided not to pay attention too much to physical applications first, but to understand how to work with them.
Ludwig worked with colleague Victor Popov. Their finding in 1967 introduced additional fields, later called Faddeev-Popov ghosts.
We were to add to it also action of some fictitious fields, which in physical processes didn't enter, but in the calculation very, very important. And so that's why they were called ghosts.
Faddeev and Popov's findings contributed to the work of Hooft and Feltman, winners of the 1999 Nobel Prize for Physics.
You should have gotten the Nobel Prize.
Well, these things are usually not discussed.
Faddeev has garnered many international awards.
So sure, I am proud.
Now a director of the Euler International Mathematical Institute, Faddeev in the 1970s was a pioneer in recognizing the importance of newly discovered solutions of non-linear partial differential equations, or solitons.
Models for which solitons were treated, we have only one space and one time, nothing else.
It took Faddeev a decade to devise a full quantum theory of multi-dimensional solitons.
In the course of this quantization, we found very important general structure, which somehow underlines all possibility of exact integrability of these equations. And this structure we called Young-Baxter equation. It led also to new mathematical structure, which is called now quantum group. It's very satisfactory when such things happen.
Nowadays, working mostly in their dacha, Faddeev, aged 74, continues work in the Yang-Mills field.
Understand what are particles which correspond to Yang-Mills field.
What is it about maths that attracts you?
It's quite attractive to have play of mind somehow. It gives you also quite internal freedom. But the main thing is there are many questions which are not answered yet. And the quest is to try to find the answer. My father told me long ago that he thinks when he was sleeping. I didn't believe him, but now I know that it's also possible.
Father was a world pioneer of homological algebra and a professional pianist. His mother, also a mathematician, named her son Ludwig for a purpose.
The reason was that my mother's dream was that her son will be conductor.
But he's always thinking of maths?
Maybe not. No.
No. He's considered a normal man, actually. He can help me, he can cook.
Ludwig and wife Anna, a physicist, met at Leningrad University.
And sometime on the fourth year, I think, we decided to go hiking in Crimea.
I invited him.
We became somehow more closer. After 2 years, we married, yes. Well, at that time was no necessity to, to officially marry, but when she began to expect daughter, then we decided that she must have father.
So you gave up your career as physicist? But I decided to
Her mother was...
stay a woman. So how would you want your work to benefit mankind ultimately?
I continue the general human tradition of making new knowledge. Skill will come with knowledge, but knowledge will not come with skills.